Good afternoon. I appreciate the opportunity to give this presentation this afternoon. This session was not set up as a debate, but I do feel just the way the flow that I am giving a case for why we are using recombinant products, certainly. These are just disclosures. So first I'd like to address the rationale for recombinant clotting factors. We will talk about safety evaluation. What have we learned from pivotal trials and post-marketing surveillance with the focus on the PUP trials? And then what are the implications of the Zipit study results? Not a question of whether the results are valid. That's really not the point of today's discussion. I accept the results for what they are in the paper. The issue really comes down to the last statement as Alok presented it. Are the study results going to change my practice, change the way I interact with patients around product selection in PUPs? There's more than 20 years of clinical trial and worldwide experience with recombinant clotting factors. Safety, did we miss one slide? There. This double clicks. Why are we using recombinant clotting factors? Clotting factor replacement products were thought that they would be safer than their plasma-derived counterparts. If you recall when recombinant DNA technology came to the forefront, it was really at the cusp and then ultimately the peak of the contamination of the plasma-derived products that patients were using. There was also the hope that consistent manufacturing and processing that was liberated from the uncertainties of securing source plasma would be a real advantage. There was potentially unlimited supply that could drive down costs of replacement therapy. This was actually a real discussion in the early days of recombinant development. I don't think we can argue with the great increase in supply that's been made available, but we would have something to say about the overall costs. Increase in the utilization of prophylaxis. I think that is unquestioned. That has been a major impact of the development of recombinant clotting factors. And then, has there been wider availability of replacement products for patients in developing countries? Again, I think if you look at the history and you look at the inflection points of getting more and more factor to the developing world, it coincides to a great degree with the availability of recombinant clotting factors. With regards to safety evaluation, we have more than 20 years of clinical trial and worldwide experience. What are you doing in a safety review? Ultimately, and this is an official statement from the FDA's website, a safety review establishes that a drug's benefits outweigh its known risks for its proposed use. So within that, with related to clotting factors, no infectious pathogen transmission, no safety signals from adverse event reporting, and no evidence of increased rate of inhibitors in previously treated patients. Now, we haven't talked about PTPs today, but it's important to recognize that PTBs have been the source of pivotal trials and to seek regulatory approval. PUPs are not part of the initial, if you like, safety evaluation for bringing products to the marketplace. Inhibitors develop in PUPs and PTPs. But inhibitors in PUPs are common, and they're considered a natural immune response to a foreign protein. It's commonly accepted that product-related immunogenicity is best assessed in PTPs. You have a patient who has been receiving a treatment product. They have exhibited, if you like, tolerance to that replacement therapy. And if they switch to an alternative product, that sets up a nice avenue to explore whether that new molecule that they're exposed to has something different around it that triggers an immune response. The wide range of inhibitors seen in any PUP series really compromises the error of statistical variation when we're evaluating these studies. This slide's already been presented to you, and we're not going to go through all these individual studies, but these are collections of both the pivotal studies that led to the registration with the federal authorities, as well as post-marketing studies in previously untreated patient population. And it really doesn't matter where you look on this slide. What should be apparent to you is really the wide variation of results across these studies. You can find some pivotal studies studies that have what seem like unusually low rates of inhibitors in pups within the recombinant population, and you can find within the plasma-derived studies what seem like unusually high inhibitor rates. So we have conflicting reports from those pivotal studies, but we also have conflicting reports from other forms of data collection, including retrospective studies, parallel cohort studies, and even meta-analyses. And the summary from these studies have shown that there is statistically higher incidence of all inhibitors, but not for high titer. If you look at the Canal studies, the Rodin, the UHAS, no statistical difference could be demonstrated in those programs. Two meta-analyses demonstrated no significant differences between the classes of plasma-derived factor VIII and classes of recombinant factor VIII, and actually suggested that factors associated with study design could impact inhibitor incidence. We had an unexpected result from one of these cohort series that a second-generation full-length recombinant factor VIII product was associated with an increased risk of inhibitor development when compared to third-generation full-length recombinant factor VIII. That's in the Rodin study. This was then similarly found in two independent hemophilia safety monitoring programs, the France COAG network and the UK experience. But two prospective registries could not demonstrate statistically significant difference, UHAS as well as a Canadian study. And with review of all these studies, methodological flaws could explain the observations. Some of the things that have been raised include the lack of a centralized laboratory and potential center effects. So one strategy was, well, let's look at a much more defined population of product classes to identify what could be evidence for a difference between at a product-specific perspective. And there was rationale to select plasma-derived factor VIII products that contain von Willebrand factor because of the protective effect of von Willebrand factor, some new insights from the immunology about how the immune system may see the factor VIII molecule, and it also seemed rational from the standpoint of some of the studies involved or that included plasma-derived factor VIII products when they were lined up and compared to the recombinant factor VIII studies. It seemed that there was what appeared to be a significant difference overall between these two product classes. But nevertheless, even from this publication, you can find within these groups what would seem like an unusually high inhibitor rate in one PUP study in plasma derived with factor VIII, and then one unusually low inhibitor rate within the recombinant group. So how do you put together the challenges from these types of studies? Well, there's non-homogeneous study designs, prospective versus retrospective. The length of observation periods are not identical through all of these studies. The frequency of inhibitor testing can vary. If you look on the study populations themselves, it's important to note whether they're including only severe populations, true less than 1%, or whether they're including some moderate phenotypes. Are the gene mutation types aligned? What ethnicities are included in the study population? Are these truly untreated patients, or is there some prior exposure? So are they minimally treated individuals? And then what's the therapy regimen that's applied? Early versus late introduction of prophylaxis, or even prophylaxis versus on-demand use? So generally, when we struggle with being able to rationalize this, we have to go to very large cohorts to try to get additional information. I think this is still the best data collection that I've been referred to from the PEDNET study group, who specifically investigated the association of inhibitors to plasma-derived factor VIII versus recombinant. They looked at VWF content as well as the type of viral inactivation method. This was 883 pups, all severe hemophilia A at less than 1%, and born within a cohort between 1990 and 2009, and they were all followed through 50 exposure days. And the results showed that the risk of all and high titer inhibitors in plasma-derived factor VIII and recombinant factor VIII was similar. And the adjusted hazard ratios there for all inhibitors and for high titer are there. And you can see that both of them, the confidence intervals, clearly cross 1. There was additionally no evidence for a difference between the low VWF concentrates compared to those that had higher VWF content, and there was no effect of different viral inactivation methods. I think this is probably as far as we can go with these types of cohort studies, and this is the data that we have. So oftentimes when we're looking for what some would call a definitive answer to a problem, we ask for a randomized controlled trial. And that's what the investigators had put forth with the Zipit study, which you've been introduced. They did decide to look at the subclass of plasma-derived factor VIII containing von Willebrand factor and compare that to recombinant factor VIII. It was prospective, it was randomized, it was controlled. 125 pups were treated, either with first, second, or third generation recombinant factor VIII, depending on the availability in the countries, and 126 pups were treated with plasma-derived factor VIII VWF. Again, as a class, they had to use whatever was available within those countries. There's no question there was a significant increase in inhibitor development demonstrated in the recombinant factor VIII treated patients compared to the plasma-derived factor VIII with VWF treated patients. But if you look at the cumulative incidence of all inhibitors, at 26.8% for the plasma-derived with factor VIII and VWF, this is one of the highest reported inhibitor rates for any trial previously involving these products. 44.5% with recombinant factor VIII, and a hazard ratio of 1.87, and a confidence interval showing statistical significance. But if you look at the cumulative incidence of high titer inhibitors, this is now 18.6% compared to 28.4%. The hazard ratio is 1.69, and the confidence interval doesn't allow us to claim statistical significance for this differential. It's possible that if this study could have continued longer, there might have been a chance to see if statistical significance would have been demonstrated, but this study had to be terminated because of the aforementioned results I showed you, questioning the role of a second-generation product that was a key part of this trial, and so the study was terminated prematurely. What that means is that this study, in contrast to the PEDNET study group data, did not allow all patients to get all the way through to the 50 exposure days. And so I will go through, I think it deserves us to have a little bit of critique here about the results for a prematurely terminated study. So the next slide. So we talked about ethnicities in one of the slides. There's difficulty extrapolating data to the European and North American populations. The majority of the patients were enrolled from Egypt, India, and Iran. We have learned a lot about risk factors and inhibitors primarily from the North American and European populations, and we don't know as much about risk factors within this group of ethnicities. A large proportion of the patients had gene mutations that were associated with an increased inhibitor risk, including null mutations. I mentioned that not all patients achieved 50 exposure days because of the early termination of the study. And interestingly, half of the patients who were treated on demand, with some receiving as few as 1 to 4 infusions per year. And Alok had mentioned the possible role that this could have in eliciting an immune response. Methodologically, there was an inhibitor cutoff of as low as 0.4 Bethesda units. This is lower than most prior studies have used. And then importantly, again, for practice change, at least within my population of patients, this study predominantly used first and second generation recombinant factor VIII. Only 16% of third generation recombinant used a third generation recombinant factor VIII. So this population actually looks very much not like my population of patients that I care for in the clinic. So what are the implications? Well, this is excellent data from the only randomized control trial comparing product classes of any nature. But it's not a head-to-head product comparison. So even choosing a product from within this Zipit study would not allow you, if you like, to choose a winner or choose which had the lowest overall risk for inhibitor formation. It was not structured to do that. All we can make a comment to is on product class. It actually does not address any of the newer recombinant products, including recent third-generation recombinant factor VIIIs, those with extended half-life, and those derived from human cell lines. It needs to be evaluated in the context of all the prior studies we evaluated today. The inhibitor risk, as always, we're weighing this against the theoretical risk of pathogenic infectious agents, but I recognize that in the forefront of what we deal with every day, inhibitors still loom large in our care practice. So medical advisory councils have issued guidance. I would direct you to the MASAC recommendation if you're interested to see how the U.S. MASAC has responded to the implications from the study. But I believe that's going to vary by country because of the availability and cost structures, etc., along the lines that Alok mentioned to us. And so I think it's really difficult for us to impose, a strategy that we may lay out within the U.S. population and suggest that that's the route that should go in every situation. So thank you very much.